video. We've got the theme for today is fluorescence microscopy, and we have Sunay Hendricks from ZOYC who will be presenting for us everything about fluorescence microscopy, different microscopes, how to choose that microscope, and some aspects of fluorescence microscopy. Thank you very much, Sunay. We appreciate your participation and your availability to us. And yes, if you are ready, please let me know. Sunay?
Hello, Lisa. Thank you for the opportunity. I am just choosing the right screen. So let's see if this is going to work. Would you please tell me as soon as you can see my screen? And now, let's see if
I can see your screen.
Yes, but it is not presenting. So sorry, let me just stop this. I want to quickly do this. Bear with me for 1 second, please.
Sure. While Sunay is preparing her presentation, can I please ask you all to switch off your videos and sound? Videos just to make sure that we have good, that there's no slowing down the connection, and then of course your microphone so that there's no disturbances. Yeah, and then if you do have a question while Sunay is talking, you're welcome to post it into the comment section. If it's something that is a general question that we think it might be beneficial for everyone to know the answer, we will try and discuss it at the end if there's time. But otherwise, I will always, I can either answer you in an email to you specifically, or we can leave it for the last day when we are actually going to discuss some questions. Right, thank you, Sunay. I assume you are ready.
I am. Can you see my full screen?
Yes.
Thanks, Lisa. All right, thank you very much for the opportunity, and thank you to all the participants for joining today. Just a little bit about myself. My name is Sunay Hendricks. I currently am the division head for research microscopy solutions at ZOYC. I've been with ZOYC now 9 years. I think during lockdown, it actually marked my 9-year anniversary at ZOYC. Before that, I did research in virology at the University of Pretoria, and majority of my research was based on microscopy with specific reference to fluorescent microscopy and confocal microscopy. So some of the topics that we will cover today, so let me just see, is I will give a brief overview of the difference between histology and fluorescence and talk about a completely different world that we enter the moment we get excited about fluorescence microscopy, and how many opportunities and applications there are, and what type of questions that we can answer with these techniques. So then we need to understand what is fluorescence, and I will then go into the different parts of the microscope that you require to do fluorescence microscopy. The main reason why I always include this in a talk is so that if you sit in front of a fluorescence microscope and for some reason you do not see what you expect to see, you can also do a little bit of the troubleshooting yourself. I will then roughly touch on different types of fluorescence, but more of this will be covered most likely in the sample prep on Friday, so it will just be a reference to it, so that we could go into then the techniques that you need to choose the right system or the right type of microscope for your type of application. So let's look at the difference between histology and fluorescence. So in normal histochemistry, you use different dyes to stay in different compartments. And typically this specific staining is so that cellular compounds or subcellular compartments with selective dyes or specific is based on the specific chemical environment of your cell type. So you have acidophilic, basophilic, and lipophilic chemical environments, and based on this, you will then see a pinkish, orangey type of dye. The main limitation of basic histochemistry is that you always have to work with fixed samples, paraffin embedded, thinly sliced. So the sample preparation is fairly routine, and you always see some sort of expected outcome when you do this type of staining. And it's quite useful for especially pathology customers or pathology so that they could then see or identify abnormalities in tissues. The information that you obtain by a certain mixture of 2 or more contrasting dyes then is, as I mentioned, typically based on the environment. So what you should see if you do a H&E stain is that you should see a dark purplish dye in the chromatin or in nuclear material, with the nucleus being purplish bluish color, and eosin, which uses sometimes orange or pink. It depends on the lighting that you use on your microscope, what type of color you will see here. But if we compare this now to fluorescence, the information that you can obtain with fluorescence is you can get a very clear separation of every single fluorescent signal and its precise location at a given time. So this opens up a whole new world of possibilities, because you're not now only limited to fixed samples, but you can also do fluorescence microscopy on live cells and even whole organisms. And then you can start to understand functional biology so much better. So let's just quickly take a look at what is fluorescence. First of all, fluorescence is just a contrast-based microscopy technique. There's nothing special to it. It's based on basic physical properties or chemical properties of and light properties of specific fluorophores that we use and utilize to bring high contrast to samples that typically have no contrast. And the another advantage of fluorescence is that it can be used to detect molecules below the resolution limit of the optical system. So if you look at this picture, the reason why I always put this in is, if you look at this picture, you should see 2 people standing in a very nice nature scene. Now, at first glance, you can see the guy with the mirror much easier, and you can possibly see him from much further distance if he has this mirror that reflects the sun. Whereas the second guy with the camouflage clothes can be more difficult to detect in a background. And this is quite a nice reference to what fluorescence really is. It is, if you don't see things with high contrast, or you're looking for a very specific protein or molecule in a whole cell or array of cells, then by adding some sort of signal to it, you can make it jump out and make it much easier to find proteins or molecules of interest in a very busy background that you might not normally see if it was there. So in fluorescence microscopy, specimens are typically treated with special reagents. Individual molecules absorb light for a very short time and then emit light again. And this emission is always shifted towards red. Okay, this shift is termed the Stokes shift after the guy who discovered it. And in fluorescence, the wavelength of the emitted light is typically between 20 and 50 nanometers longer than the absorbed exciting light. So what you need to take home from this slide is we need a high energy, low wavelength to excite molecules in my sample. Once the molecules absorb enough energy, they want to get rid of it again, and it would start emitting light with a lower, longer frequency. And the difference between these 2 wavelengths, as I've mentioned, is called the Stokes shift. So when you want to do fluorescence microscopy, you always have to check what the excitation wavelength of your specific dye of interest is and what the emission wavelength of your dye of interest is. And this is also if you're looking at experimental design and you want to use multiple different dyes for multi-channel fluorescence, then this is also something that you need to consider for overlapping dyes, whether it is excitation or emission wavelengths. So these are the things that you need to consider when setting up your experiment in the beginning as well. So just a different way of explaining this, you need high energy short wavelengths to cause excitation of a sample, and if there are any molecules that can absorb the energy, you should see lower energy, longer wavelength light coming from your sample again. And this is just the physics. The diagram at the bottom is just the physics behind that. So specific molecules would take up energy, and as they want to go back to a lower energy state, they would emit it in the form of light. Okay, and there's just it's the same diagram, just explaining it in a little bit more detail. So let's see now, if we look at what parts of the microscope we need to consider for doing fluorescence. The very first thing is you need a specific light source to give enough energy so that you can create enough energy for exciting the molecules in your sample of interest. So typically in this example we have a mercury arc lamp, and this is a broad spectrum white light source. In the microscope that you see, the diagram that I have here, the lamp would always be on the same side of the objective. So on the image here I've got a compound microscope, and the light source is on the top half of the microscope, on the same half of the microscope as the objective. If you go to a microscope where you're working with an inverted microscope, typically used for cell culture, and the objectives sit at the bottom of the microscope, the light source would always be at the bottom on the same side as where you find the objectives. Okay. So at the back of the microscope we have the lamp house, or the light source. In this case, we have a mercury arc lamp, which gives off all the different types of potential wavelengths that you're going to use for excitation. As the light travels from the lamp through your beam path of the microscope, it would reach a filter cube. Let me just see if I can find a laser pointer. There we go. All right, then as the light travels through your microscope beam path, you will get to a filter cube. A filter cube inside your microscope consists of an excitation filter, a dichroic mirror, and an emission filter. The excitation filter allows very specific wavelengths to pass through it, and it would block out wavelengths of your specific choosing. For example, in this particular diagram, we are blocking out light that is longer wavelengths than the blue light or UV light. Only UV light is allowed to pass through the excitation filter. UV light is then reflected onto the sample by the dichroic mirror. The dichroic mirror would reflect light shorter than UV, and anything longer than UV light would be allowed to pass through the dichroic mirror, also depending on the angle where it comes from. So UV light is then reflected down onto the sample, and excitation occurs of the dyes. Light of longer wavelengths then come out of the sample, and as it reaches the dichroic mirror, because it is longer wavelengths, lower energy, longer wavelengths, it is allowed to pass through the dichroic mirror. It then reaches the emission filter at the other side of the cube, which again then chooses which wavelengths of light could go through to the detector, and in this case it is your eye. So if we look at this and we have a specific filter cube that allows you to see GFP in your sample, then that filter cube would allow UV light to go to the sample, excitation occurs, and green light is then emitted, which we then see down the microscope. Okay. Now, if we look at different types of light sources, we can have different options. All microscope providers give you the option of mercury pressure lamps, LED. In the ZOYC case, we have a special light source called Calibri, or a xenon-based lamp house. In our case, it's an HXP 120 light source. So let's just look at some of the advantages and disadvantages of each one of these. So how do you choose, if you're looking for a fluorescence microscope, what do you need to consider to choose the right lamp source? So normal filament lamps are not suitable as light sources for fluorescence microscopy because the glowing metal filaments convert most of their electrical energy into red or invisible infrared light, which means that the wavelengths are too long. So because it is long wavelengths, it's low energy, and therefore it's not enough to create excitation of specific dyes or molecules. As I say, their fluorescence requires intensive shortwave light. So your high pressure mercury lamp is time-tried light source for fluorescence because it is a broad spectrum, it gives off a discrete light spectrum, which would give you specific excitation of almost all available commercial dyes. Some of the disadvantage of working with mercury pressure lamps is that these lamps need to warm up before you can start using them. So you typically have to switch on a mercury lamp and wait for anywhere between 20 and 30 minutes to build up enough pressure so that it gives you quite a strong, consistent lamp light from the lamp. Once you've worked with this, with it, you also have to wait for it to cool down before you can switch it on again. So there is always I'd say at least a 30-minute waiting before you can start working for it to warm up. And there's also at least a 30-minute cool down before you can switch it on again. These lamps also have a specific lifetime. So the typical recommendation is that you swap the lamps between 200 and 300 hours. So you always will see that there is typically some sort of record book where you have to fill in how many hours you've used the mercury lamp for. And this is so that you can track the amount of hours that the lamp has been, or the usage. So even though it can still give off light, we do recommend that you change it between 200 and 300 hours, which makes mercury lamps quite costly because you have to change them so often. They also have to be disposed of in a very specific manner. Another light source which is very popular because of the low running cost of these is LED. So LEDs are quasi-monochromatic emissions, very specific. If you look at current LED technology, and this is a fairly old slide, they have a long lifetime beyond 10,000 hours. I think we specify now 62,000 hours on our Colibri light source. So anything between 60 and 80,000 work hours before these start to lose energy. These light sources, so our current light sources, also self-calibrating, which means that you don't have to worry, it's always going to give you the same strength compared to the other Colibri diodes that you have in your lamp house. They have ultra short response time. You can switch it on and immediately start working. And there's no heat radiation, no vibration. It's integrated into your microscope and you can use different combinations. For these type of light sources, you also don't need an excitation filter because these are practically monochromatic emission. You don't need to block out any alternative wavelengths because these light sources give off a specific wavelength of light already. So your filter cubes are also designed slightly different. As I mentioned, it does not have the excitation filter. If you're not quite sure and you start off with fluorescence research, we typically recommend going for an HXP lamp. These also an external white light source, a separate box that is coupled to your microscope and can be coupled to any fluorescence microscope. These quite nicely fill the gap in green and yellow ranges, even though the latest LED technology now also have developed quite significantly to include to cover more of the dyes in the green and yellow ranges. HXP is quite tried and tested for that. It's very fast shutter. You can also just switch it on and switch it off. There's no waiting periods. The lifespan of a xenon-based lamp is anywhere between 600 and 2,000 hours. So they're also a lot more stable than HPOs. So typically, if a customer doesn't know what type of dyes they're going to use, or they're only starting out with fluorescence, this is typically what we would recommend you start off with, because there's no limitations. You can simply just add different filter cubes as your research develops. All right. I've mentioned already, I've spoken and explained the principle of a fluorescent cube. The only thing that I'd like to just add here is it is quite important to know, as I've said, the excitation and emission wavelengths of the dyes or the molecules of interest that you want to work with, because it can be a limitation if you choose the wrong filter cube, or you cannot give that type of information to the person that helps you set up the system. You can lose a lot of light, because if your excitation or emission filter blocks out any of the wavelengths of the light of your molecule of interest, this can create in a very dim sample or no fluorescence at all. So these are already some of the things that you can check if you don't see the type of signal that you expect down your fluorescence microscope. All right, that's just a different diagram, again, to show it in a different way. I would say, I think Anya explained it very well in her presentation on Monday, but I can never stress how important choosing the right objective is for when you want to do fluorescence. So some of the things that should be considered is if you are working with dyes that have very low or very high wavelengths, then you need to ensure that the type of objective that you're going to use are suitable for high light transmission values into the UV range for excitation, and then into the far red range for emission, if that is something that, if those are the ranges that you're interested in doing your research. Some of the other considerations is that a lot of your lenses, filters, or even some of the immersion liquids that you use can exhibit autofluorescence, which will create in a lot of background noise. So sometimes if you need to do troubleshooting, and I see it often that people use any type of immersion oil when they do fluorescence microscopy, and then they don't understand why all of a sudden there is so much light coming from the background of the sample. So you do get specific immersion oil which has been treated to be fluorescence free. This is typically what you will also find in the core facility, and this of course comes at a cost. But if you want to take Nobel Prize winning images, then it is worthwhile spending that little bit of extra money to get the best type of image for your publication, especially if you want to do any type of image analysis afterwards. Then very important, fluorescence excited or coming from your sample is radiated or emitted in all different directions. So you always want to work with objectives with very high numerical aperture, because the larger the opening of your objective, the more of the stray light you can grab from the sample. And that is why there's always the perception that if you go to a higher magnification, you will always get a brighter signal when you look down the fluorescence microscope. And it is because the objective can grab more of the radiated light that's coming from your sample. So if you want to get very good fluorescence images, always choose high numerical aperture objectives. And this picture, which I think Anya showed on Monday as well, explains basically that. So if you have an objective with a larger front opening or a higher numerical aperture, you would be able to grab a lot more of the light coming from your sample than light in any other way that would be missed by the objective because it's radiated in all different directions. So, just one comment that I'd like to make, which we often find when customers consider doing fluorescence microscopy. First of all, you need to understand that microscopy on its own is already a very much a light-starved technique. The amount of light and energy that you have to put in is always going to be greater than the amount of light and energy that you get out of it. So it's, I'm not going to worry about that. That is a very specific application. But I've mentioned in the beginning that fluorescence is just an imaging technique. So what is important when you work with fluorescence is to consider that we always want to grab all the information that comes from the sample, and every photon coming from the sample is sacred. So when it comes to imaging or detecting any of the light that comes from our sample, we need to look at specific cameras that has been designed for this. Now, normal DSLR cameras, or the ones that you are used to, the type that you find in your cell phone, or these old snap cameras, I don't know how many people still use it with the current available technology that we have in our cell phones, but these type of cameras are made for very light-rich environments. So they typically have very many pixels that are very small. They have fast readout configurations, and these are typically a CMOS design. You get very high digital resolution and very poor sensitivity and dynamic range, mainly because you don't need it. If you're going to take family pictures on a Sunday at a picnic outside, then if it's a beautiful sunny day, you're going to get very nice images, and you can actually see the resolution because you have very high resolution cameras because of the amount of pixels. However, microscope cameras are designed for light-starved situations. That is why they already have very large pixels. Now, because we design the pixels to be much larger so that they have very high sensitivity and high dynamic range, you will always find that microscope cameras have a lower amount of megapixels. I often get the question, yes, but this is only a 5 megapixel camera. Yes, but that is what is suitable and needed for a microscope, because there are different things that come into play. As mentioned, sensitivity, dynamic range, and sometimes speed and so on. We also use typically very sensitive CCD sensors for best signal to noise ratios when we want to image fluorescence. Currently, our best top of the range fluorescence camera in the ZOYC portfolio is a 2 megapixel monochrome, in other words, black and white camera that is used for fluorescence microscopy. And that is our top of the range camera. Excellent for microscopy applications. ZOYC also offers a special dedicated talk just on microscopy camera technology, how many megapixels are enough. And if anybody is interested in that, we could also make that available to you at some stage. All right, so just a quick touch on the different types of fluorescence that you can use to study fluorescence microscopy without going into too much detail, because a lot of this will be covered in the sample prep for Friday as well. So some of the biggest challenges that we face is when we work with samples with a lot of autofluorescence. So this can be very useful as well, permitted that you design your experiment in a clever manner. Alternatively, you can use immunohistochemistry or immunofluorescence, as it is also called. These comes with also very special sample prep techniques, and there's some considerations, and I'll just touch on one or two things. And then lots of fun, easy to use, are when you label proteins directly to your protein or labeled proteins which have a fluorescent molecule coupled to the protein of interest, so that you can study these in live cells as well. Alternatively, live cell probes, which would then also attach to specific cell compartments or molecules of interest, so that you could see these in live cells. Autofluorescence, this is typically where you see a lot of this in plant material, and this can cause a lot of problems, because the diagram that I showed down here shows if you just use UV excitation, you can see a whole range of your cell organelles light up, which can cause challenges, especially if you are interested in something that is labeled with a GFP protein or a molecule that is has a specific wavelength in the blue-green ranges. Yeah. These are just some examples of autofluorescence, but some of these examples also use dyes of interest. I think so in this example, they also use YFP to study. Now, this is something that I will cover in the sample prep or in one of the discussions on Friday. We will talk about multi-channel fluorescence imaging and also spectral unmixing with the confocal microscope. The best system for looking at autofluorescence samples is your confocal, because the confocal detector allows you to separate these signals coming from the different organelles or dyes or areas of interest. Immunofluorescence, nice, because this can be used on fixed or live samples. As I said, here you have a primary antibody that binds to your target protein or organelle in your cell, and typically a secondary fluorescent antibody which binds to the first. Just a note here, you can also get direct labeling. So direct labeling is if your primary antibody has the fluorophore or the tag attached to it. This makes the protocol, I'd say, only about 1 or 2 hours shorter, but it does have an influence on the amount of fluorescence. And the reason for that is every primary antibody, you can only bind to a specific set amount of antigen sites on your target tissue. But every primary antibody has between 4 and 6 binding sites for a secondary antibody, which means that if you do double labeling, or if you use a primary and a secondary antibody, then you can increase your fluorescence signal between 4 and 6 times. And as I mentioned, we're working with light staff techniques, so if you can do anything to enhance the signal, it should be a consideration in your sample prep. Of course, there's many ways in a protocol where you can do some fault finding, and I'm sure that some of this can be discussed in some of the groups on Friday. Then you can also have labeled proteins or coupled with probes. These are just some examples. So the most well-known example is the green fluorescent protein, or GFP. And over time, as people have worked on it, they've seen that if they mutate the serine 65 in GFP, it leads to enhanced GFP, which is just a very much brighter version, which I think is sort of the standard that is used now. They've also seen that if you put different aromatic amino acids at the position 66, you can get a whole rainbow of different colors, and also use this for different techniques. Then there are also some labeled proteins which can be used for different colors. These are some of them are derived from coral, other sea corals, and one example is, for example, DS red. Without going into too much detail, just touching on this, so multi-fluorescence imaging gives a very big visualization complexity. The biggest advantage is that typically we are looking at biological material with no or very limited contrast by nature. But by introducing fluorescent labels, we can look at live or fixed samples, and you can see the relationships of different and multiple targets, even protein-protein interactions and protein organelle interactions in cells or tissues, or even now where biology is going, whole organisms. So here are some examples of what fluorescence can be used for. So you can have fluorescent dyes that are coupled to antibodies. You can express fluorescent proteins. You can have ion-sensitive dyes, dyes selecting for subcellular structure, voltage-dependent dyes, or even active uptake of fluorescent particles to look at different cell dynamics. And these are just some examples of, on the left-hand side, bacterial colony made in this beautiful sunset with expressing different fluorescent proteins. So it can also, I just like this because it's quite playful and it shows how much fun fluorescence microscopy can be. And on the right hand side you have a brain bow, which is a transgenic mouse expressing a combination of different fluorescent proteins. Some more examples. So here are some examples where you can see spatial and temporal labeling of specific proteins and with reference to plant material. You can see the different development stages as well. Now, in order to choose the right system for your application, I quickly want to go over one or two principles of fluorescence microscopy. First thing is, why do we need special techniques? So forgive the fact that this comes out of a presentation with regards to a specific component, which I will not discuss today. But why do we need to look at different special techniques to image fluorescence microscopy? And this comes down to this point spread function of a microscope. So what does that mean? If I put any, let's call it a micro dot, or a fluorescent point structure, there is my sample. If I put this under the microscope, then unfortunately, the right-hand picture is the reality of what I will see. And so you see a very bright dot from the middle, and then these concentric like light rings that are coming out of my sample. Now, the good thing is, because we understand and we know the quality of the optics, the quality of the light path, and really all the components that go into the microscope and everything that influences the formation of this image, we can still calculate what the resolution of that point is in the microscope. So how we calculate the lateral resolution of my system is based on a specific constant, and that times the wavelength of the light that I use divided by the numerical aperture of the objective. So again, it all comes back to the quality of the objective that I use. So the higher the numerical aperture, the better the resolution. Now, we all know that we don't just work in 2 dimensions. And the moment that we add a 3rd dimension, because that is what biology is. It's 3-dimensional cells, organelles, or structures. The moment we add a 3rd dimension, this whole idea just becomes one big fat challenge. So we luckily can still define this. So the axial resolution also has a very specific equation that is coupled to that. Right. But now, if we look at this, the image of a single point in 3 dimensions is no longer a point the moment that I put it under a microscope. It becomes a very complex 3D diffraction pattern. So how do we address this? I've mentioned to you that in biology, we're typically interested in things in 3 dimensions, not just the axial resolution is important, but 3 dimensions. So how do we take 3D images in a fluorescence microscope? Well, we take images at different focal planes to create a 3D image stack that allows us then to see in 3 dimensions. Right, so we take images at different focal planes and put them together. Okay, but if we do that, this is typical what we see in a normal wide field fluorescence microscope. So the sample that I have here is a Drosophila embryo, and here you can see, just because of the point spread function of fluorescence, the spatial intensity distribution causes a lot of out of focus light. So the structures that are actually in the focal plane, or the structures that I'm interested in the sample, becomes completely blurred by light above and below the focal plane. In other words, the out of focus light causes this haze, and this is no fault of anything apart from the basic principles of the point spread function of the fluorescence molecules, or then the microscope. So how can we make these images better? We want to do optical sections. Now there's different ways to improve image quality through optical sections, and this is what I want to talk to you about now. So that allows us, if we start working with optical sections, then it allows us to take this image and really block out anything that is not in the optical slice, so that we can improve contrast and resolution of microscopic images and really see what lies behind the out of focus blur. Right. I'm now going into a different presentation, so just bear with me 1 second so I can stop sharing this one and open up the next. Up until now, are there any questions so far? Hannes, you have a question. You can actually unmute you if you want to just ask me.
No, it was a mistake.
Oh, okay. Then at least thank you for raising your hand and seeing. Is there any other questions? Anybody, any comments?
So now there is a comment. It's a question on Saeed says, if I'm working on rocks for maceral composition, what type of fluorescence microscope would I need? I'm not sure if that is something you can answer now, or should we leave it for the end?
Let's leave that for the end. Saeed, I would actually like to find out quite a lot more. First of all, the first question that I would ask you is, are these rocks autofluorescent? And then you can use any fluorescent microscope. It depends on really what you want to see on these rocks. So it becomes a whole discussion of what your sample is, what type of fluorescence do you expect to see, and what information do you want to get out of it.
Yeah, but I want to identify the different types of materials like cont, fluorescent, vitrinite. Then from there, I'll be able to define the portfolio.
I'm not sure if anyone can hear you, Saeed. I think from my side it's very unclear, the audio. Maybe you can put that into the comment section.
The organic materials in the rock. There are different types of organic materials that we have in the rock. Choline, sporinite, vitrinite, alginite, and the rest. So the fluorine, like fluorine. So if I'm working
Saeed, stop please. I really think it would be better if you type out the question fully, and then at the end, Sinai can answer the question with more information, but we can really not hear the audio from your side. So if you can type it into the comment section, then maybe Sinai can answer that at the end. Would that be okay for you? Okay. I'm going to give over to Sinai again.
All right, Saeed, I'll gladly take this discussion with you offline as well. But yes, let's have a discussion if there's some time at the end, and then we can have a chat about that. Or alternatively, we can set up a offline discussion as well. But I'll gladly have a discussion with you, and we can talk through the type of information that you want. All right. Can you see my screen?
Yes, I can.
All right. Okay. So this second part of the discussion is not just purely for living cells. The idea of this second part of this presentation is really to look at what do I need to consider. So really say that it refers back, it comes back to your type of sample, your sample as well. What do you want to answer? Which type of questions do you want to answer? And then once I know that, I can now consider what type of different techniques are available for myself. But first, before we can go on, let me see. Right. So I'm quite excited about this picture. So before we can talk about new imaging technologies, let's just take a look into the past. And let's see the very first system that was launched by Carl Zeiss in the early 50s, mid-50s. And this system is called the microcinematograph or something. I don't even know how to pronounce this. But if you look at this image of the system, there's quite a couple of familiar components already to this microscope. And I'm going to point them out to you. So here we have a microscope. There's some optical system. So there's the microscope inside. You probably have the objectives there. You have this incubation chamber around it to keep the temperature stable. And then this whole thing is coupled to this big device, cinematograph or whatever it is, how it's pronounced, that will then allow you to take a movie of what you see under the microscope. And this was where live cell imaging started for Zeiss at least. Now I'm going to show you one of the very first movies that was taken with a system in its very first development stage. So in the 40s, people were already interested to see how cells work and what happens inside cells. So I get quite excited and emotional because I'm a biologist, and when I see these even early footage of cells dividing, I get this very warm, fuzzy feeling in my stomach because this is exciting type of research. And this is really, if we could have done it 50, 60 years ago already, but with some advances now, we're much further into what is possible with microscopy now. So here is just an example of what we see now. So a lot of things have changed, but we can still look at the same processes. So what you see here in this example is a cell going through mitosis and the chromosomes being separated by the mitotic spindles. One of the things that has dramatically changed is that now we can label specifically the macromolecules we are interested in by using fluorescent proteins, which have been developed in the mid-90s. And this, as I've mentioned already, have changed the world of microscopy in the most impressive ways. Here I have another example. One of the most interesting features of fluorescent proteins is that they can be used to label any type of sample, from bacteria to animal cells. And as we can see here in plant cells, in this example of the microtubules, which is labeled in the root cells of Arabidopsis thaliana, allowing to visualize processes in the cells that don't look very active on the outside. Another example is we can look at complete organisms, which we can now image with one or even more specific labels that allow you to observe almost any biological process you have in mind. For example, here in this embryonic development of Drosophila, again, the microtubules are labeled, which allow us to observe that cell divisions are not happening uniformly throughout the embryo, but some areas have more divisions than others. So, in this talk, I would first like to define some of the key parameters that you should consider when you are trying to decide on your imaging. And then we will discuss different examples of samples and look into the best microscopy technique to use. But first things first, when you are imaging, you want to consider signal to noise. So, signal to noise is effectively how much stronger your sample signal is compared to your background noise. As you can see in this image, the signal to noise is progressively higher, becomes progressively higher as we go from A to D, so that eventually we can see Charles Darwin. But hand in hand with this, we also need to, so a second factor that is very tightly associated with signal to noise is gentleness. And that is because often when you try to increase the signal to noise in your image, you would often increase the laser power if you're working on a confocal microscope, or the light intensity if you work with a fluorescence microscope. But that can lead not only to photobleaching, but also phototoxicity. And it has time and time again been published that excessive light exposure can cause damage to your sample. So the signal to noise, together with gentleness, is what we are going to refer to as system sensitivity. Right. So if you have a sample that bleaches fast or live cells that can be susceptible to phototoxicity, then you want a system that is very sensitive. And this is one of the first considerations. Next, and my apologies, I just want to move my mouse over here. All right. The second imaging parameter to note is how quickly the process is that you want to capture. Okay, here in this example, you can see the beating of a cilia in the ependymal cells lining the brain ventricles. Now, this typically requires very fast imaging. I think this image was taken at 143 frames per second. However, it is not always essential to get a system with very high speed if your sample doesn't justify that. If you're working with fixed cells or fixed samples or stuff that isn't moving at all, or not moving, bacteria dividing only once in every 20 minutes does not necessarily need a very fast imaging system. But if you want to do calcium imaging in muscle cells, then you would typically require very high speed to capture the signaling events. The third imaging parameter is then the depth of your structure. For instance, are you looking at the top layers of an organ, or, like in this case, are you looking at the depths of a brain and trying to capture the length of the axons? Tissues have an inherent ability to scatter light, and the deeper that you go into your sample, the more light scattering you need to consider. And then imaging techniques and sample preparation become a very strong discussion as well. So the sample thickness or the size of your sample should also be considered if you're looking at a whole brain slice or a single cell, and whether you're looking at stuff at the cell walls versus very deep into your sample. The fourth imaging parameter is resolution. Resolution is defined, and I think Anya explained this on Monday as well. Resolution is defined as the minimum distance between two structures that would still allow you to see them as two separate objects. This practically means that if you're looking at a structure, like in this example of the Drosophila neuromuscular junction, if that structure is beyond the resolution capability of your objective, then you will typically just see a fluorescence blob or a glomerular structure. But the moment you increase the resolution, you can see the actual donut shape of these neuromuscular junctions. Now, you also, talking about this again, have to consider whether the type of sample that you look at have these very fine structures and whether you need that to answer the types of questions that you want. I once worked with a group that was looking at how dough rises. So this was food science, and they wanted to look at different types of yeasts for cookie dough. Now, giving them a super resolution system is going to be of absolute no benefit, because their sample does not justify adding resolution. There, they would more likely look at something that is sensitive, because they would much rather get as much of the signal as possible, or see how these cells grow over time or something like that. All right. The fifth image parameter, which I will not go into too much detail, is how many samples you need to work with, and really considering the throughput of your work. Because if you're looking at a single well versus looking at 384 well plate, it would take a completely different amount of time. And whether you need the information from all 384 wells is also a different discussion. So if you want to do high throughput, for example, typical example here is people that do a lot of pharmaceutical work or drug development, they want to typically see a lot of information with a snapshot. Then you need to consider different techniques with automation as well. Okay, and lastly, the sixth and last parameter you need to consider is the size of your sample. So, do you want to look at cultured cells, whole zebrafish organisms, or live mice, and look at a complete mouse brain? And here you can see that things have moved towards more systems biology. One of the biggest hot topics, and most of the inquiries that we work with currently, are people that do organoid or zebrafish type research. So your size of your sample also needs to be one of the biggest considerations to choose the right system. Okay, so for the sake of simplicity in this talk, I'm going to define samples as thin, medium, or large. And then we can consider them more or less equivalent to maybe a cell, an organoid, or a whole zebrafish. Okay. Right, so if we place the imaging parameters we discussed on a grid, then we can use this type of plot to create, let's say, the perfect system that excels in each one of these parameters. And we can then score them accordingly. So this would be then a visual representation to see what type of system would give you the best of each one of these parameters based on the type of research. So let's typically look at, for example, if you want to look at something with high sensitivity and high speed. When you plan your experiments, you need to consider then all of these parameters and then decide, well, which of these 6 considerations are non-negotiables and which ones can I be more flexible on? And then based on that, you can then choose the right system or microscope that is suitable to your application. It is important to note that there can be overlaps on some of the parameters. Right, I just want to switch this off quickly. Okay, so some of the technologies and so on, there will always be an overlap. But let's first look at, let's just look at some cells. All right. And if you just want to look at some cells and you want to be real quick, then you can use a Widefield microscope. So what is a Widefield microscope? That is a normal fluorescence microscope that you find in a lot of labs on at any university campus. Okay, in a Widefield microscope, the image contains light from the focus area there, as well as above and below the focal plane. All right, so in a Widefield system would always have a lot of blur, which we've shown in the previous samples. If you work with a confocal microscope, then you can avoid a lot of the background signal and therefore generate much better image contrast. Okay, and then, as I've mentioned, we can do different Z stacks throughout different focal planes. If you use a Widefield microscope, then there is no optical sectioning. If you use a confocal microscope, then we block out a lot of the out of focus fluorescence. So what is the implication? let's just look at a wide field system, but that we also use then software to create a better image resolution. So what we have here is an example of living dividing cells imaged with very gentle illumination at a very high speed with a normal fluorescence monochrome camera that allows you to capture the whole image field simultaneously. As you can see, the image is beautiful on the left-hand side, but there's a lot of out of focus light. Now, one way to improve the image is to do deconvolution. But what is deconvolution? It is a way to improve your image by processing it after you've acquired it. And in fact, it's a technique that has been very well established for a very long time. Deconvolution can help you resolve your image better, and it works best when you have a lot of light, for example, as is the case in a wide field microscope. Wide field microscopes have a lot of advantages, especially if you want to look at live cell images or sometimes even fixed samples. It is very gentle illumination, all right, with, for example, the Calibri or LED illumination, light illumination that I've mentioned earlier. And because we use very sensitive CCD-based sensors in our cameras, you also get a very high signal to noise when it comes to the sensitivity of these cameras. So it's very quick and your full image field is captured. Okay, what you should consider is the sample has to be very thin to make use of these advantages. For example, 1 cell layer is good. Okay. Moving on, you can also do live cell imaging. Here you can see deconvolution can help you resolve microtubule structures better, and our ZOYC software offers fully integrated deconvolution, very easy workflows. It gives you 4 options, you choose one, and because we've moved away from we are moving GPU-based deconvolution, it is extremely fast as well and very easy to use. Okay. If we look at wide field and deconvolution, we can then say that if you use this, then it is a very sensitive because of the CCD camera. It is high, it can be high throughput. So if you have a motorized scanning stage, you can snap the information in a multi-well plate really quickly. It is very fast because you capture the whole field of view at the same time. And if you use deconvolution, you also increase the resolution. However, the optimal size, sample size should be very thin, and you're not going to go in very deep. All right. So let's say you want to look at a little bit more of a versatile system. Right. Sorry, let's see if this moves. All right. Yeah, this is when you want to use a confocal microscope. Okay, this parameter combination will strongly support the use of confocal microscopy, which is one of the most versatile imaging platforms available. Confocal microscopy allows you to optically section very thick samples, to separate multiple fluorophores, and to do photomanipulation experiments. This technique is great for multicolor and thick samples. It gives you high resolution, and you can also get quite good depth penetration. And here are just some examples of images that have been taken with a confocal microscope. You can also do live cell imaging, and here you can see different time-lapse videos. So if I just go into a little bit more detail, before we go into detail, sorry. Yeah, we can also do 4D imaging. So this is an example of the CNS in a cockroach. So the LSM allows us to acquire very large volumes of data, multiple labels at very high resolution. And then with special visualization software, the Arriva software, you can then separate all the signals and really look at all the different signals at very high resolution. I've mentioned spectral imaging and sample unmixing. So this is real-time separation of 4 fluorophores in the presence of chlorophyll. When you want to work with, for example, any plant material, you always struggle with a strong overlap, as I've mentioned before. So when you want to image a sample labeled by multiple fluorophores, it is inevitable that you're going to have some labels that overlap. But the LSM can quickly solve this issue with spectral unmixing. And in this example, you can see that we can separate up to 4 signals simultaneously. You can look at photo conversion. So if we just start these videos here, you could see the red dyes appear, or that the sample changes because of interaction of different samples. So this is also called fret, right? Where you have the moment that your proteins start interacting, there is a photoconversion or a difference in color, and you can measure this in real time. On the left-hand side, so this is a fret experiment. On the right-hand side, this is a FRAP experiment, fluorescence recovery after photo bleaching. So here we bleach an area of interest, and then we track how long it takes for the fluorescence signal to move from adjacent areas back into that area. So here you can actually look at protein movement over time. And this is all possible with a fluorescence LSM confocal microscope. So you can also, the exact precision of the LSM that allows you to do quantitative measurements of protein dynamics and interactions through fluorescence correlation spectroscopy as well. In a nutshell, fluorescence correlation spectroscopy is monitoring the movement of a single fluorescent molecule through the excitation volume, and the measurement you require can help you determine the molecule's quantity and movement dynamics, as well as their association with other molecules. And we do have a special module called FCS for the confocal microscope that you can do this as well. The popularity of the method has been significantly increasing in recent years, with more and more groups using fluorescence correlation spectroscopy to further characterize their model systems. Here you can see an example of monitoring TF diffusion dynamics using FCS in combination with photoactivatable GFP. Using FCS helped the researchers identify that the diffusion dynamics can vary for different TF and stem cell lineages, giving them insight into the behavior of single protein molecules. So, confocal is probably your most diverse sample. So I won't go into detail. Next week, Monday, I think we're going to talk about super resolution, and then I will talk about all the different things that we have developed with our new LSM 9 family to help push really into the sensitivity, into the throughput, going much deeper, increasing the speed, and really increasing the resolution of confocal microscopy as well. As a base, confocal is your most flexible type of technique for the majority of samples. So let's say you want to also now look at very deep samples. Then you will do multi-photon microscopy. So what practically this means is that with a 2-photon or a multi-photon excitation, you can see image structures at greater depths with less light scattering, allowing you to visualize much deeper structures. For example, in the case of motor neurons innervating in the mouse muscle. You can also look at calcium imaging, and this is depth coding. So this is how deep you go into the sample. And let's just start this. See, there you go. So you can see every time there is a flare, you can see calcium imaging very deep into your zebra embryo fish. I think this is the spinal cord. So with 2 photon, you can see much deeper. It is high throughput, and you can still see sensitivity. It's not necessarily as fast, and the reason for that is you're going into so many deeper levels of your sample. So you're always going to have some sort of sacrifice. Super resolution. I think Lisa will cover this early next week, but just so that you know, if this is a normal Widefield system, then we can get with normal structured illumination microscopy or SR-SIM, we can get down to resolution of 120 nanometer, which is basically double that you can find on any fluorescence microscope. And you can also do live cell imaging with this at very high resolution, depending on whether your samples need that. You can also do super resolution with multiple different colors. Here you can see the beautifully resolved mitochondrial membranes and microtubule growth as the cytoskeleton reorganizes. So if you look at SR-SIM, then the system gives you a really good, very high resolution. So it's fair throughput, not necessarily deep tissue imaging, but it's quite fast, excellent resolution, and quite sensitive. Now, what if you want to have a sensitive, fast system, but you look at very, very big organisms, so quite large sample size? So in this example, we look at a light sheet microscope without going into too much of the details. So when you are imaging with a confocal microscope, the entire sample is exposed to light while you're collecting a Z-stack. Whereas when imaging with a light sheet, we only shine light through the focal plane that you are currently imaging, also the focal plane that your imaging is exposed to. The effect of this expose might be negligible in thin samples, but when imaging large samples and doing continuous stacking slices, the increase of this over time can lead to bleaching and phototoxicity. So if you want to look at embryo development or specific events inside whole organisms, then this technique is much more sensitive because you only illuminate what it is that you're imaging at any given specific time. So yeah, you could see the amount of slices that is necessary and the amount of time that you want to necessarily image this. And this is then what is going to cause, if you have less photo damage, you can view your samples for much longer. It means that you can also then track the development over a longer period of time. So due to the design of the light sheets microscope, the sample is suspended in water or other medium and is moved through the sheet of light as you image it. And the selection of the aqueous solution is very advantageous, and water is a very stable medium due to its high specific heat capacity. The other advantage, so this is just an example, the other advantage of using a light sheet system is you can rotate your sample, so you can move it up and down, in and out, closer or further from the objective, and you can rotate it at 360. So you get a very large volume of information at a fair speed. You can also image. So here's whole embryo development over 6 hours in this example of a flower beetle embryo. Very, very gentle imaging. Okay, I've mentioned that we could rotate the sample 360, which allows you to get multiple views and acquire a lot of data. Then I just have a couple of examples. This is a symbiotic marine anemone and emerging coral systems. So to sum up, a light sheet system would give you excellent combination of speed and sensitivity and depth, and would suit larger samples. And it is very advantageous because you could rotate your sample so you get a lot of information, which in my opinion makes it quite large throughput because you can see a whole organism, but you cannot see multiple organisms. So the take-home message is that the light microscopes are very diverse and that they all have different unique strengths and limitations. The key to success for you to consider is your imaging parameters and to decide what type of questions you want to answer, and based on that, decide which type of system will fulfill your needs, considering that there is always some sort of payoff between the different techniques. If you would like more information on any of the systems or any of the techniques, I will share with Lisa. We have a website, the Microscopy Insights Hub, and there's a lot of information regarding the systems and also a lot of additional application webinars that we have published on our website. With that, I would like to thank you for your attention.
Hi there, Sona. So Lisa was kicked off due to load shedding. Would you like to tackle Sahid's question, or would you like to speak to him on another Teams meeting?
Sahid, if it is okay, I think because of the line, because the line that was a bit bad, I think we can have an offline discussion. So Anya is also in the meeting. I'm going to ask you to share your information with Anya, or we will get it from Lisa, and we will give you a call and have a discussion about your sample and see. I did find that you want to look at organic material inside these rocks. But we need to just have a discussion, and then we can look at some more considerations there. All right.
Great. And then Lisa had a question. She said, I have a question on depth of focus on a confocal microscope. Is there a way to know the specific limit in the depth of focus into a sample for a specific objective?
Yes. So I can quickly see if I can open it, and I can share this useful site with you. Give me 1 second. I'm just opening it up on my laptop, and then I'll share. And this anybody has access to this. So just 1 second. I'm just opening it up, and then I will share my screen. One second, I just want to open up a specific objective, and then I can show you what we have. Okay, thank you for your patience. Let me quickly see if I can share this. Is my screen sharing? So if you go to our Carl Zeiss shop, or you can just search for the Zeiss Micro shop. It will take you typically to the German website, or any of the websites. We have, if you go to tools, we have an objective assistant. And if you click on that, it allows you to search for any particular objective that you have on your system. If you then go to that objective and you go to the full description and the characteristics, you can see all the information on any objective in our portfolio. So here it gives you the magnification, numerical aperture, the free working distance, the type of thread and so on, the field of view that you see with this and the parfocal length of the system. What the suitable applications for it. So this objective is really, really good for fluorescence microscopy, for multi-channel, ultraviolet and infrared. It can also be used for bright field microscopy and specifically differential interference contrast. And here it would give you all the different information. You can also then get the transmission curves and the accessories, transmission curves, objective classes and descriptions. Regarding the depth the sample penetration depth on a confocal microscope, there if you work on a 63 or 100 times, you're probably not going to get any deeper than, I would say, about 50 micron into a sample. Then it's already very, very deep. Okay, so I think that the standard limitation is between 40 and 50 microns on a confocal microscope. Anything deeper, that is if you don't use any special sample preparation. If you work with spheroids and organoids and you then do sample preparation and you do clearing of your sample, then you're also going to limit light scattering and then you can see deeper with your confocal microscope. So we have groups that do confocal microscopy with spheroids that are up to I'd say about 500 microns thick, and they get very, very good information. But I'd say you limit to, I'd say, around 50 micron on confocal. Then you need to look at probably a multi-photon system.
Okay. We have another question from Saeed. He asked, does the deconvolution use an algorithm or does it require an extra feature?
So our deconvolution module already is programmed with specific algorithm for you, and it gives you 4 different options are based on the most popular algorithms. So you can then choose whether you want quick and dirty. Based on your sample, you can choose any one of these, but you don't have to it doesn't require anything special. All you need is the Zen software and the deconvolution module. And it would already do that. You can also program it yourself. So you can also then manually do it. But currently the algorithm is so good that you don't need to do that. And it's extremely easy to use. You literally click one of 4 options and it does the deconvolution for you.
Okay. And then there was a question from Jubilani. I'm not sure what he's trying to ask. He can maybe activate his mic, but he said, hi, would you advise me to use all different light microscopy instruments? This I would consider if I want a better understanding of my sample, especially if I'm doing a novel project.
Jabalani, this is yeah, so thanks. I see Anya have answered you a little bit there. We would actually like to take this discussion and find out what is your sample, and then it's much easier if we can ask you a couple of questions and then say, all right, well, what is it? What are the research questions that you want to answer? And then recommend what type of different microscopes you can use based on what it is that you want to answer. So if you want to see how fast things move, whether you have a new drug target or whatever it is, we can then recommend based on that. Otherwise, I would always refer somebody to the most versatile tool, which you have in the SAF anyway, which is the confocal microscope. So yes, but please send an email to Anya if you would like us to have a more in-depth discussion about your particular project.
Then I have a question about titrations. My background is flow cytometry, so there's quite a procedure to follow to determine in flow cytometry what is your optimal volume to use for a conjugated antibody. Yeah. How do you do that in microscopy? How do you know?
It's the same.
Oh, it's the same.
There's always, no, so I would always recommend, so it depends on if you're going to buy a commercially available antibody, then they typically recommend a primary antibody of 1 in 100 or so on. Yes. Then they would typically recommend a suitable titration for your typical application. Typical for primary antibody, it's 1 in 100. I can't remember. It depends on the secondary antibody and the specificity. But that could be 1 in 200 to 1 in 1,000, depending on the specificity of it. So these things, I would recommend optimization. And this is something that I think we will also cover on sample prep for Friday, is how do you prepare your sample? What optimizations are needed and which things to consider when you actually want to do fluorescence microscopy?
Just another question from me. If you do fluorescence microscopy, if you do batches, say 5 batches in a week, your settings need to be the same, right? From Monday to Friday, or in batches, you need to keep the settings the same, right? Like
if it's the same sample, and if you want to do any quantitative analysis afterwards, I would always recommend using the same settings. But then compared to what? So if you want to do fluorescence microscopy, there should always be the relevant control samples. You should always have a positive control, a negative control, always as a sample that you've only treated with secondary antibody so that you could determine the specificity of your non-specific secondary antibody binding, etc. So it's very, very important that you have the right controls, first of all, to determine the baseline, and then again, if you just want pretty pictures, you don't have to have the same settings. Then you're going to change your settings to get the most beautiful picture for your publication. But if it is a case where you want to do quantitative analysis, image analysis afterwards, then I would recommend, yes, that you keep your settings the same on each one of the batches so that you can compare apples with apples.
Okay. But this is not applicable to light microscopy, right?
No.
It doesn't really play a role.
No,
or settings, keeping settings constant.
No. I guess, again, it comes down to even if you want to do in light microscopy, so normal bright field microscopy, yes. If you want to do any quantitative analysis, it is important to at least keep once you've found out what is sufficient for imaging, then you should keep most of the light intensity and exposure time of the camera the same if you want to compare a lot of samples with each other.
Okay. All right, is there any more questions from anybody? Not. All right. So, is you. All right, so I'm going to finish off now. It seems that somebody wants to ask a question, but we can't hear properly. So thank you very much, Sonia, for your presentation. It was very informative. And also thank you for your time, for setting your time aside to help us today with our presentations.
I really appreciate the invitation. Have a great day.
Thank you. You too. Bye, everyone.
Bye.
Thank you.
Pleasure.